Good afternoon, everyone. My name is Francesco Albano, and actually I'm a researcher for the University Federico II of Napoli. I will thank the organizers for inviting me and for giving me the opportunity to share with you those data about a project I worked for a couple of years ago, and is titled ZFP36L2 Role in Thyroid Functionality. Congenital hypothyroidism is a state of inadequate thyroid hormone production at birth. It may involve both thyroid dysgenesis or dysarthrogenesis, and defect of thyroid development or position, or thyroid hormone biosynthesis. Different gene mutation can cause congenital hypothyroidism. Thyroid dysgenesis has been primarily associated with mutation in genes such as those encoding the TSH receptor, FOXY1, and PAX8. Dysarthrogenesis has been associated with mutation in sodium iodide cotransporters, thyroid peroxidase, dual oxidase 2 or dual oxidase maturation factor 2. Although PAX8 knockout, NKX 2.1 knockout and FOXY1 knockout mice revealed the importance of those genes in thyroid development, few germline mutations have been detected in the humans and in corresponding genes in human patients. Papers published in past years on this issue suggest that mechanisms leading to congenital thyroid dysfunction are heterogeneous and involve several signaling pathways, including Notch1. Transcriptional mechanisms controlling thyroid development and function, including epigenetic aspects, have been deeply investigated. Although the regulation of mRNA stability is also an important mechanism regulating differential gene expression, particularly in the development process, its role in congenital hypothyroidism has been neglected. The role of two RNA binding proteins, AOF1 and UR1, have been investigated in thyroid carcinogenesis, but not in congenital hypothyroidism. Recently, one or more hairs have been found in silico in the 3' UTR region of several important thyroid transcripts, including NIS, TSH receptor mRNAs, and their transcription regulator NKX 2.1 and PAX8. Furthermore, the regulation of NIS mRNA stability has been reported in response to iodine, both in vivo and in vitro. The tristetraproline family members ZFP36, ZFP36L1, and ZFP36L2 are CCCH tandem zinc finger proteins that promote decay of the mRNA targets. TTP family proteins were found to act as regulators of apoptosis and are involved in cell differentiation. Gene targeting studies in mice define distinct roles for the ZFP36 family members, for example, revealing that ZFP36L2 is a key regulator of hematopoiesis. It also plays a role in female fertility, and its inactivation has been associated with the impairment of lipid metabolism. To date, its involvement in thyroid development and function has not been reported. We have previously observed that ZFP36L2 mRNA was enriched in mouse thyroid buds and decreased in model of thyroid carcinogenesis. Furthermore, ZFP36L2 expression was reduced in thyrocytes exposed to environmental stressors. Here we report that ZFP36L2 gene inactivation in vivo and in vitro affects thyroid function and thyrocyte survival by mechanisms involving Notch1 signaling. We started the analysis of the role of ZFP36L2 in thyroid development and function by using a complete knockout mouse model of ZFP36L2. As previously reported, complete ZFP36L2 knockout mice died approximately 3 weeks of birth before weaning. ELISA measurement of circulating thyroid hormones in serum samples did not show any major difference in circulating free T4 between ZFP36L2 knockout and control mice. At postnatal day 7, CFT4 levels were reduced in ZFP36L2 knockout mice at postnatal day 21. Possibly because the thyroid hormones supplied by the mother through lactation were no longer sufficient. Since no males were available, we evaluated only females. Thyroids appeared to develop normally and were not ectopic in ZFP36L2 knockout mice at PND 21. Hematoxylin and eosin staining of the section of the thyroid gland showed a trend toward the decrease in the number of follicles together with an increased presence of fat in the thyroid of ZFP36L2 knockout mice versus control. To investigate the role of ZFP36L2 in thyroid function at a later life stage, we developed mice on a C57 black 6 stain background with thyroid-specific inactivation of ZFP36L2. These mice were obtained by crossing the ZFP36L2 floxate mice with the mice expressing PAX8 allele, driving gene inactivation mainly in the thyroid and kidney. Circulating free T4 and T3 were determined by ELISA at postnatal day 30, 60 and 90. No major difference in circulating T4 were detected in males compared to control alternators at any sample time. The conditional knockout mice, the female mice, manifested a trend toward a decrease in circulating free T4 levels at 1 month, at 2 months, and the level of circulating free T4 and T3 were significantly reduced at 3 months. Concurrently with the hormone levels, the conditional knockout females had a reduced body weight compared to controls. Considering those data and the fact that females are more susceptible to hypothyroidism, only this sex was further investigated. Hematoxylin and eosin staining of thyroid section did not demonstrate any major defect, although slightly smaller follicles were detected in the conditional ZFP36L2 knockout. To more completely characterize the thyroid phenotype, thyrocyte proliferation and apoptosis were investigated. No major differences in proliferation levels by Ki67 staining were observed between the conditional ZFP36L2 knockout and control females. Furthermore, in order to detect the possible presence of apoptosis, we performed tunnel assay staining. In this way, we detected a slight decrease of apoptotic nuclei in thyroids for conditional knockout females versus control. These data were associated with an increase in the ratio of BAX over BCL2 mRNA. Then we investigated thyroid functionality by determining the expression levels of thyroid-specific transcripts, like NIS, TZ, and TPO, and transcript for the thyroid-enriched transcriptional factor PAX8 and KX2.1 by RT-qPCR. This data demonstrated a reduction of PAX8, NKX 2.1, NIS, thyroglobulin, and thyroperoxidase transcript in the conditional knockout for ZFP36L2 females. The decrease in PAX8 and NIS levels were also demonstrated by Western blotting. Collectively, the data show evidence of hypothyroidism in the conditional knockout females, and that the inactivation of ZFP36L2 in the thyroid affects the function of the gland in more ways than its development and morphology, except for the slight increase in apoptosis detected in the thyroid-specific ZFP36L2 knockout females. To investigate the molecular mechanism underlying the effects of ZFP36L2 inactivation in thyrocytes, we generated a cellular model by knocking out ZFP36L2 in immortalized right thyroid cell, FRTL5. This is a well-characterized in vitro model, widely used to study thyroid function, since it retains the ability to concentrate iodine, produce thyroglobulin, and to respond to TSH stimulation. The ZFP36L2 knockout FRTL5 cells were obtained by lentiviral delivery of the CRISPR-Cas9 components, whereas control cells were infected with an empty vector. We determined the levels of the ZFP36L2 mRNA and protein, both in the bulk culture and in selected clones. We chose them to work on the bulk culture as being more representative of the heterogeneity found in vivo model. ZFP36L2 knockout cells did not show any major changes in cellular morphology versus empty vector cells, since transcript playing a role in the regulation of the cell cycle and apoptosis are targeted by ZFP36L2 in other cell types. We tested both cellular activities. MTTSAs did not show any major differences in the proliferation of exponentially growing empty vector and knockout cells. Despite that, we found an increased expression of P21, suggesting an impairment of the cell cycle. Furthermore, the increased duration of PAX-BCL2 transcripts pointed to a possible increase of apoptosis. Molecular characterization of thyroid-specific or enriched transcript and proteins in exponentially growing empty vector and knockout cell many agreed with what we found in mice. Indeed, PAX8, NKX 2.1 and NIS mRNAs were significantly reduced, as were their encoded protein in knockout cells. In contrast, no differences were observed for thyroglobulin and thyroperoxidase in the knockout cells, or for FOXY1 TTF2. As member of the ZFP36 family may have redundant function, we analyzed the cellular content of ZFP36L1 and ZFP36 in the knockout cells. Levels of the ZFP36L1 were reduced in knockout cells, whereas the ZFP36 mRNA showed a slight trend toward the decrease. Overall, the data suggests a role for ZFP36L2 in thyrocyte functionality in rat immortalized thyrocytes. Since TSH stimulates both cellular proliferation and expression of thyroid-specific or enriched genes in FRTL5 cells, we investigated whether the loss of ZFP36L2 could affect those cells' response to TSH. To test this, empty vector and the knockout cell were deprived of TSH by growing them in a medium without TSH for 3 days. Such a procedure resulted in a FRTL5 cell synchronization in G0G1 phase. After starvation, the cells were stimulated by adding a fresh complete growing medium for 24 and 48 hours. We choose these time points since FRTL5 doubling time is reported to be 36 hours. We test whether ZFP36L2 levels could be directly regulated by TSH, and we found that ZFP36L2 protein levels were reduced after TSH starvation and increased after TSH stimulation. Interestingly, the levels of ZFP36L2 protein seem to be increased in starved knockout cells versus empty vector. Same behavior was observed with ZFP36L1. Furthermore, TSH appeared to promote ZFP36L2 protein phosphorylation since treatment of the extract with calf intestinal phosphatase resulted in the appearance of a single band of an immunoreactive protein. To confirm TSH signaling, we analyzed NIS protein level in knockout and in empty vector cells. NIS was downregulated in starved knockout cells compared to controls, and the increase after TSH stimulation was lower, and it was delayed in knockout cells. Overall, this data suggests that the response to SAH was impaired in absence of ZFP36L2. Since TSH controls both proliferation and survival of thyrocytes, we investigated both processes in knockout cells. Cell counts showed a reduced number of cells after 48 hours of TSH stimulation, and FACS analysis of the cell cycle showed an increased G2M population in knockout cells, suggesting both at 48 and 24 hours, suggesting a faster exit of the knockout cells from the G0, G1 phase. The distribution in different phases of the cell cycle of the knockout cells was similar at 24 hours and 48 hours, indicating a longer duration G2M phase. We found consistently an increase of P21 content in knockout cells after starvation and TSH stimulation. Increases in the phospho ERK1 and 2 were detected in all conditions in knockout cells, despite the overall reduction of the ERK1-2 proteins. Analysis of the P38 MAP kinase activation showed an increase of its phosphorylation, already detectable in the starved knockout cells, and even more evident after 24 hours of TSH stimulation. Interestingly, the level of total P38 protein showed a reduction in knockout cells. The increased phosphorylation of P38 in knockout cells was already apparent after a shorter period of TSH stimulation. Another potential mechanism by which ZFP36L2 could modulate the levels of thyroid-specific transcript was described in T lymphoblastic leukemia, in which ZFP36L2 and L1 were able to modulate the cellular content of Notch1 mRNA and protein in a signaling pathway that is involved in thyrocyte function. We assayed the levels of the Notch1 gene in our experimental setting in vitro and in vivo. Notch1 gene expression was increased in exponentially growing knockout cells, as well as in TSH-starved cells. Furthermore, TSH stimulation increased its level in empty vector after 48 hours, whereas it had a little effect in knockout cells. This was also in agreement with the reduced ERA binding activity of ZFP36L2 when phosphorylated. This result was confirmed in thyroids from conditional knockout mice. Indeed, an increase in Notch1 gene expression was detected in non-conditional knockout mice and was further supported by the induction of HES1, a direct Notch1 target. Notch1 and HES1 are positive regulators of the expression of thyroid-specific genes that decrease following ZFP36L2 gene inactivation. We looked for other Notch1 targets that could act as transcriptional suppressors, since ED4, a transcriptional repressor, is a target of Notch1. and has been reported as negative regulator of PAX8 transcriptional activity, we attempted to verify the increase in our model, and we found by RT-qPCR that levels of the ED4 transcript were increasing in knockout versus empty vector cell, as well as in thyroid from conditional knockout mice versus controls. Collectively, our data suggests that ZFP36L2 knockout or reduction might exert its effect by deregulating Notch1 signaling and by promoting an increase of S1 and ED4 levels in thyrocytes. This imbalance might damage thyroid function and survival and condition its development and differentiation of cells. Thank you everyone for the patience and for the attention.